We're now moving to the last talk in this morning session by Kang Yu from the ETH in Zurich. He will present about the capabilities of the field phenotyping platform FIP, demonstrated by characterization of the time series of canopy cover in wheat as related to G-by-E interaction. I think that's already using math of the time.
Thanks a lot for reading a long title. I think we might save 2 minutes time to go for lunch if you don't read the long title. So I would like to thank the IPPN organizing committee choose my talk to present here. And I'm a postdoc in the crop science group working at ETH Zurich. So today I'm going to show some preliminary results from our field phenotyping platform, so the ETH FIP platform. So why we are doing phenotyping? So especially when we are doing field phenotyping, because we want to know how plants respond or adapt to changing environment. So especially the highly fluctuated field environments. So based on this fundamental question we are asking, and in last 4 to 5 years, ETH Zurich has established this unique cable suspended field phenotyping platform, so FIP. So this is the area of the FIP. It covers 1 hectare area in the experimental station of ETH Zurich at Lintal Echken. And this platform has 4 pools building, so on the 4 corners of this area. So these 4 towers, under the 4 towers, there are 4 winches. So in the winches, in the winch houses, 4 winches moving the sensor head by pulling or releasing the cables. And we have a control room, so basically the operator sitting in the control room, so monitor how the FIP works in the field. And in this field we have 6 lots, so every year we have 2 lots for wheat experiment. And to freely orientate the sensor head, we have this pan and a tilt drive. Does that work? Sorry, that's not okay. Yeah, okay. So we have this pan tilt drive, so that allows us to watch the plants from 3 dimensions freely. And recently, so after I have already submitted this abstract, we have submitted 2 manuscripts. So I'm going to talk one of the preliminary results of the manuscript about the canopy cover, and one of my colleagues, Lucas Kronenberg, going to show in his poster about the plant height measurements using laser scanner. So we have developed a canopy cover segmentation pipeline based on machine learning. So it classifies the illumination conditions in the field, because in the field the light conditions not like in the well-controlled greenhouse or other conditions that you can have a relatively constant light condition in the field. You have to account for the variability of light differences. So we developed this pipeline that allows us to classify the illumination differences. And next, we also using a machine learning method doing pixel level based classification that allow us to segment the ground cover. And so this is the annotation of our field with 6 lots, and we see the really huge field variability, so across the time of measurements. Sorry, should get closer. Okay, so you have seen the huge variability. So based on 3 years measurements and analyzing 60,000 of images of a near-infrared camera, so they are made on 100 days in 3 years. So these images are taken in 700 plots. So just Dr. Thomas has mentioned this 300 European which genotypes. So I think this is a rather subset of that population. So we did imaging every 3 to 4 days, so during the 3 years. So before last year, the FIP could not really automatically running. And that's the reason we also did hand measurements in 2014 and 15. Okay, this is the increase of canopy cover against growing degree days. So I will going to show the results mainly focused on the early growth phase. So we see the quite linear relationship of canopy cover related to growing degree days, because growing degree is the total amount of heat that plants needs to reach a certain progress or phase of the growth. And we also see interesting that at some phase, the canopy cover is negatively related to VPD, so the vapor pressure deficit. Okay, we see that different environmental factors affect the growth of canopy cover. So how can we dissect this effect? How can we understand the G by E interactions? So then we fit a 3D plane, so against GDD and VPD. So based on this, we can get 3 traits, or say dynamic trait. So the intercept to the canopy cover, the slope to on the axis of GDD, and the slope on the axis of VPD. And we did the linear mixed modeling to correct the field variability, as we see the huge variability of the field. And based on the BLAPs, so the best linear unbiased predictions, we calculate 3 years heritability. So unfortunately, at the moment, it's not so high, although it's very high in individual years. I think this is the effect of the intrinsic canopy structure of different genotypes. And by doing similarly on a subset of 29 genotypes of Swiss varieties, and we see a significant increase of the heritability across the 3 years. So that's because we have more replicates for these 29 genotypes, and the different growth phase might also have a different degree of influence on these 300 genotypes. And based on the results, so we have the dynamic traits of slopes and intercepts. And we did the GWAS analysis. We could find significant marked traits associations on the chromosome 2B, for example. The A is based on canopy cover growth rate in 1 year, and the B is based on plant height growth rate of 1 year in 2015. So we're going to do more analysis across 3 years, try to find if we can get the same markers for these 3 dynamic traits. Okay, then I come to summary. So based on preliminary results and the running of last year of the FIP, we can say that FIP at the moment allows for tracking crop growth throughout the growing season and to track the growth dynamics. And the dynamic traits can be extracted from the time series of traits. And we might get insights by doing this kind of modeling of G by E interactions. And growth is hugely affected by the growth phase and also different environmental factors. And we will do more analysis of the growth rate of canopy cover, plant height, and other traits that can be made by the FIP in the future. And some outlook when we are doing field phenotyping. And the FIP-like platforms, so in the field, can be used as a reference platform for nowadays for UAVs, but UAVs plant phenotyping studies. And sensor and data fusion is going to be a challenge, also opportunity for field phenotyping. And markers or genotypes identification to really understand how crops respond and adapt to climate change, that's also important. And by understanding this, that will allow us to design optimum crops for the future. Okay, challenges and opportunities. So we see the huge variability of the field. So we need really a proper measurement or imaging setup when we are doing imaging-based phenotyping. And high throughput image analysis and quality control of the analysis is also important. And multiple years and say doing the same experiment in different sites will help us to understand the G by E more properly. And I would rather interpret this as opportunities rather than challenges. So we also need advanced data analysis, interpretation, and fusion methods for field phenotyping. Okay, why we think there are opportunities? So, as we expected in the controlled environments or in the field, often we want a trait that can be nicely monitored or analyzed throughout the growing season or at different phases of the growth. But often there something happens in the field that we could not expect before we doing experiments. For example, lodging. Okay, that's the opportunities we are facing at the moment, I think. And I also would like to draw attention to come to Lucas Cronenberg's poster. And I would like to thank the FIP and the Crops Science Group at ETH. Thank you very much. And I would like to show you shortly the FIP video. And during this time, you can ask me questions.
All right.
So this is the FIP sensor head. We call it dolly. So it moves smoothly in the field. So it can go to any position of this 1 hectare area. We also have the weather station installed in the field. So this is the control room.
All right.
Right.
Any questions?
Not yet. So this is a this is a plot wise measurements of the 300 genotypes. So
this is the chair.
Oh, sorry.
All right. Do we have any questions, Ralph? Sorry, Michelle.
I was wondering, is it winter wheat? So
Yes.
grown in the true winter,
Yeah.
before winter? Okay. So what was the relationship you found between your ground cover rates, phenotypes, and yield in those winter wheat scenarios?
Yes, thanks a lot. I think this is a very good question. So at the moment, we could not really say that the canopy cover, this canopy cover trait is related to the yield of different genotypes. We have the yield recently, analyzed the data, but we haven't seen the results yet. But I guess there will be some relationship between the relative growth rate of the canopy cover and the final yield. This is my guess. Matthew, please.
Over here.
So you mentioned that on this platform you fly drones. So can you see any discrepancy or perhaps high hidden effect of drones, etc.? Because to some extent you have the truth data and you can compare the drones with it.
Yes, I do see the possibility. So because with this FIP, we don't have the problem of normal drones that have the downwheeling wind. Say when we are trying to fly 3 to 5 meters, normally at the lowest, I would say. But with the FIP, we could fly two.
The question is, do you see drawback of drones? Because you have the tools and you can compare your tools with the drones.
Yeah, I would not say it's drawback because with drones you have more flexibility during the.
If you compare the drone data with the data that you collect with the FIP of the same canopies, what is it that you don't get with the drones that you get with the FIP?
Okay. Thanks a lot. Yeah, thanks a lot. Yeah. So one example is that laser scanning data. We know that laser scanners are much heavier, so it's not easy to have a big drone to carry the laser scanner in the field. So I think also dangerous. Although it's possible at the moment to do the 3D imaging, 3D reconstruction to get the height, but I have seen some reports that the resolution is, or the precision is not so high as laser scanning measurements. Yeah.
All right, one quick question and quick answer.
I will be very quick. Just the question is about when you have environment interference, for example, foggy day or when it's raining or when it's very shining, and then your soil will looking very different. Will that impact your 3D laser data points, and how you actually denoise these data at all?
Yes, sure. So when we have raindrops, so the ultrasonic detector of the FIP dolly will detect the raindrops. So in this case, we cannot really freely driving the FIP dolly in the field for safety purpose. So when it really say big raindrops coming, we are not really able to driving the FIP. But for laser scanning measurements, I think it's still possible when we have very few drops, because normally it stays at the location, scanning 1 or 2 hours or several minutes. But we can drive relatively for a long time to get to the position we want to go.
All right, thank you very much. We'll have to finish the discussion at this point. If there are more questions, you can catch up with the speakers later. So please join me in thanking all the speakers of this. Now we've got about 15 minutes before you get lunch and more oxygen. One thing we would like to do is, if people have suggestions about workshops for the Thursday, so if you have the idea that you really want to catch up with a whole group of colleagues and discuss this one subject that you won't have enough time during coffee or lunch, then this would be an opportunity to sort of suggest any topics that people might want to discuss. Feel free to. None at this point. I'm not sure, Thomas, anything about time course, QTL, mapping? This is mainly my self-interest here. Any thoughts at this point, or do you need more time to think? More oxygen, all right. Then I think we can, or do you have anything else?
Yeah, I mean, there are more questions from earlier talk from any of the questions from any of the talks, any of the speakers, or comments.
Yes, actually, hello, I'm Aaron from the University of Ghent, and I wanted to ask about this from the second last speaker. When you take the working PS2 efficiency, you image from the top, and I wonder what is the light distribution at different heights, because then you can image leaves that are at the top and image that are at the bottom, and they probably are receiving different amount of light. So then the PS2 efficiency that you are working is not comparable between those two leaves. How you take that into account?
Yeah, that's a very good point. Actually, the LED panel, the fluorocarbon panel, is equipped with optics, so the radiation angle is about 30 degrees. So that will keep a fairly homogeneous light distribution, although a slight shift in intensity is inevitable. However, these shifts are very minor, and there is an area of about 40 centimeters height where we get very comparable intensities, and we checked very comparable PS2 efficiencies. Maize is not affected by these moderate changes in the intensity. Tobacco is. So it depends on the species whether in the range of distances you may have an effect that you need to consider in the analysis or not. But we have very carefully checked that, and we can go through these check data.
Yeah, but that check, it's also with the plant or is with the empty chamber?
This is the empty chamber, and we measured the plants in different distances to the panel, the very same plant, and measured the operating efficiency. And we could show that it does not change depending on the distance for maize, but it does change for tobacco.
Okay.
So it depends on the plant species, and of course on the range that you are. Whether you're within 10 centimeters differences, no problem at all. If you're in 40 or more, might be a problem, depending on the species.
Okay, thank you.
Don't run away, we've got another question for you.
Okay.
Thomas, Trevor Garnett from the Plant Accelerator in Adelaide. You found big differences in that efficiency, like 30%, wasn't it? Have you
photosystem to operating efficiency? Yeah.
So have you looked at that further, like validated or investigated that further? I mean, it's a huge difference in them.
We have validated. Astrid has grown several times the same plants, and we found back and validated that these differences occur. So it's genetically inherited. And actually, we were surprised that we were not seeing a good correlation between the size of the plant and these parameters, the photosystem to efficiency parameters. So there are big plants with low efficiency, there are small plants with high efficiency, and vice versa in that collection.
We're just analyzing this more detail at the moment.
It's not a question for you, sorry, but I have asked one already. I have a general question, and I'm not sure what we want actually. We have seen very simple phenomobile that we can push. We have seen automatic ones, and I'm just wondering, what do we really want? One thing is it's great to have cheap Phenomobile. The other thing is that we have to push it all the day, so we might have social problems in the units. So no, but I would be interested during coffee breaks or anything to perhaps better refine the needs, which are probably not the same in countries, sorry to say that, in which the manpower is cheap, or other type of countries. But I would like to discuss that one way or another anytime.
Was that a comment directly to that? No? Question? So do you want to workshop that on Thursday in more detail, or?
I don't know. It's just a question. It can be, if you think it's a good.
Could you capture it in a title?
You realize that you're being
price versus utility, cheap phenotyping versus capability of people to handle cheap things that need a lot of manpower, and probably So
I think Bernie would probably call it fit for purpose.
But I think perhaps we don't want to have this discussion now.
No, I think that's a good topic for Thursday, definitely. So sophistication versus cost accessibility or something like
Yeah, but it's sophisticated, and the cost of non-sophistication is the cost of the manpower.
Okay, yeah. So, yeah, cost-benefit analysis of high throughput phenotyping.
I have a comment about this. I think it all comes in one word, and that's cost efficiency, or maybe two words, whatever it is. Cost efficiency. Yeah, cost efficiency is the one, and it covers all the concerns that you have.
Can you speak into the
No, that's fine, but I think we need to. It's a good idea.
So I think there is plenty of opportunity for discussions. Okay, I think there was one question over there. Matthew.
Where?
My question was.
Wait, let's use the
Hold on.
mic. You.
My question is for Tracy, sorry. So I am curious about how many plants can you phenotype for this pipe platform, the one that phenotype the roots in the adult plants. How many plants can you do it by, in one day, or how many pictures do you take per day of each one? How do you do it?
So you meant specifically the big pots?
The ones that are in the pipes, in the big pots.
Okay, yeah, so those scans take about 4 hours, and you can section the scan. So if you're just interested in, say, the top 20 centimetres, that might just take an hour. But the generally, X-ray CT, you can do scans from about 5 minutes to an hour. But the large pots take 4 hours just because you scan them in sections, like 4 20-centimetre sections.
So do you take pictures every day on every plant to have? So
Depends
how
what the
many
biological
plants?
question is. Like if you were looking at, say, the diurnal effect of water uptake, you might take scans every couple of minutes. But if you're just scanning once a day, you just take a scan once a day. The biggest time constraint will be the image analysis. So it might only take 5-10 minutes to do a scan, but then it could take days to get the information out that you require.
Thank you.
There's a mic.
Last week, CIMMYT hosted a meeting of 20 gene bank managers from the CG system and national programs. And at this meeting, 6 of those gene bank managers are attending. So one topic that we could discuss on Thursday would be the characterization of broad types of genetic resources, broad diversity in genetic resources. So rather than high throughput, perhaps high genotypic diversity.
All right. Good suggestion. I think we've got a comment from Uli.
Does it work? Yeah. We yesterday talked about seed phenotyping or seed bank phenotyping work group in IPPN, and there is also a DivSeq seed bank phenotyping group, which I'm chairing. So if you want to do that, we can talk about it.
The interesting challenge of that is it's definitely high throughput, but it's the huge diversity in phenology, morphology that you get with exotic materials. So that's a really interesting topic. Yeah.
All right, I think we'll wrap up at this point. Matthew hopefully noted down the suggestions for workshops. Just one last housekeeping before we break for lunch. Anyone speaking in the afternoon session, could you please upload your talks in the break? There won't be change of laptops, so please come and make sure that your presentation is working.
We've got another topic here.
Oh, sorry. Okay, one last topic before we run out of oxygen.
Just on behalf of the root phenotyping committee, we'd like to suggest for Michelle's keynote to discuss root phenotyping traits and how they might be successful, if people would want to talk about, to carry on the discussions from yesterday morning.
What, you've had a whole morning and you still...
We want more, yeah. Well, that was the feedback. The feedback was we want more. We love roots. We want to talk more about roots.
You need a whole day.
All right. I think we all need oxygen. We'll see you back at 2.